and the recording has started, so you guys can pick back up at Euandrena.
All right, I'm going to flip to Mike's guide, and I can pull specimens, and we can, oh, that's the male guide. We want to go to the, which we might, oh no, yeah. Oh, no, I just have to, yeah,
that's a female.
I just have to make it a little bit bigger, I think. Yes, 150% would be good. Oops. There we go. That's better. Yeah, if we have time, I'm kind of thinking that we probably won't, but we could get into Mike's male key too. But I think we ended, as Claire mentioned, at Rhacandrena, and now we're jumping into some ugly business. So, Mike, take it away.
Yeah, but we said this last week, but subgenera Rhacandrena, Euandrena, and Thysandrena, which we'll cover, lack any overall distinctive morphology, at least the way I see it. They're all kind of stripped down, nothing fancy about them, and as a result, they kind of, superficially look the same. And I know we covered Rhacandrena last week, but just want to mention it. There are 3 species in the East. One of them is a specialist on sumac, one's a specialist on Amorpha, and the other one, I'm not sure because I don't see enough of it to really know. So that would be Brevipalpus is on sumac, Krigiana is on Amorpha, and Robertsoni. I don't know, Sam, what you think, but I just don't see it enough to really know.
Yeah, I think it's on. I think it's on sumac.
Okay.
Wait. Well, let me back up. Let's say rosaceous plants earlier in the spring. Okay. I'd have to go back and review that myself.
Yeah.
Because its records are earlier than Brevipalpus.
Yeah. Well, one of the things those 3 females have in common that Euandrena and Thysandrena don't, and it's subtle, but the dorsal propydium, the triangle, is more coarsely sculptured in those 3, the Rhacandrenas, than in Euandrena and Thysandrena. But it's not like Rhacandrena. I mean, it's not really reticulate, but it's comparatively, when you look at Rhacandrena and look at a Euandrena or Thysandrena, the propydium is rougher. Let's put it that way. So that
I think of it as sort of like Nasonii.
Yeah. Minutely wrinkled, kind of, rather than smooth. It's definitely not smooth.
Yeah.
Well, that's good.
So here's Robertsoni. Maybe I can go one more level. Oops, there we go. Yeah, like Mike was saying, kind of a uniform, rough, all filled, not a lot of change from the base to the tip, topography.
And then the tergites are punctured, but I mean, it's, as you can see right there, they're weak, and there are not many of them. But in Euandrena and Thysandrena, there are hardly any punctures at all. But again, it's comparative. You really have to see all 3 groups to understand the slight differences.
And I forget whether, is it Robertsoni that has, you can see sort of the tessellation, the fingerprints, the reptilian part, more than Brevipalpus. But that often can really obscure, particularly if the specimen is a little filmy, it can obscure the pits, because the pits are fine.
Yeah, that's a good image. That's great.
Yeah, and you can also see here's gook all over here, and that's going to obscure all the characters, like here in between. I'm hiding it, but you can see the pitting in it among the tessellations, and then you have, as is often the case, just splotches of stuff on the surface, which also points out why it is good, why you want good, clean specimens if you can get them.
So that's Rhacandrena. And you did see the fascia there too, but that's not some of the other species in these other two subgenera have enough indications of fascia that it's, and plus it can wear and be messed up, so it's hard to rely on that too much. So yeah, what comes next? Yeah, Euandrena. So that couplet below Rhacandrena, again, is, I think we talked about this last week. I kind of apologize for it. It's kind of a lot of different characters all thrown in there, if and then type of thing. So Euandrena and the Thysandrena that are coming next, certain groups of them are distinctive. So with the Euandrena, there is a group of species that have pointed galea and sharply pointed. I mean, not these are, we'd all agree that they're sharply pointed if you can see them. But if you can, I mean, that right away, you know, it's this group of Euandrena that in the east there are 3 species, and we Polemonia specialist, a Phacelia specialist, and then a hydrophyllum specialist. And so they stand out, and they also sometimes have a little bit of slight bluish sheen to them, depending, and again, it depends probably on how they were treated post-collecting. You know, gunk can mess that up. But if they're good specimens, you can kind of see that slightly bluish, and it's subtle, but it usually it's distinctive enough that it's not just all dark. So that's a subgroup. Yeah,
I'm going to pull a Pulmoni while you're talking, Mike. So
okay. So that's a subgroup of Euandrena, but all 3 of those occur in the east. And the only other Euandrena that are in the east are northern, algida and is it W scripta? Yeah, and they're really obscure. They're all dark, and I mean, in the Midwest, they don't occur unless you're up the upper part of the Great Lakes region, usually. And I don't know about they're probably in the mountains, I would imagine. But they have rounded galea. They're not bluish, and so the algida comes out below. Well, let's see what else.
Hey, Mike.
Yep. Yeah.
What were the other species by Pulmoni? I can't seem to.
Phacelia,
I guess I didn't keep any of.
Phacelia. Yeah, or Gerania.
Yep.
Which is the biggest and the neatest looking, really. And that's in a way, it's an unfortunate species name because it's not a geranium specialist. It's a specialist on hydrophyllum, waterleaf. But it's one of the cooler Andrena. Another unique thing about Geranii is the propodial scopa. Propodial scope is it's essentially absent. And you remember in some of the other earlier Andrenas, the scopa on the propodium is like a basket, or it's very distinctive. But with Geranii, it's not there. There's hair, but that's all. They're not modified in any kind of a way. That's another unusual feature of that species. Did you find one?
Yeah, and I'm gonna show it here. All 3 of those species, at least where we are around, but very uncommon. We just don't have, like, for example, we don't have large amounts of Phacelia, and Geranii, we don't see that one hardly ever. And same with Pulmonia. We don't have any records. I thought I kept some from Delaware, where it showed up on Jacob's Ladder at Mount Cuba.
Yeah, they're common in the Midwest. In music forest kind of situations, in stream banks, but what I would call natural habitats. They're not weedy Andrenas at all. They're
right.
You pretty much have to go to nice quality habitats to find them.
Even in this out of focus, you can see the sheen of blue-green metallics for this Phacelia.
Is that from Maryland? Sam?
I think it's from Tennessee or North Carolina, actually. Yeah. I don't know that I may not even have any records. I have it sort of surrounding Maryland and West Virginia and Pennsylvania, but I don't think we have any records for Maryland. So let me see if we can see the pointed galea on this.
And a lot of times, even if you don't spread the mandibles or pull the mouth parts after you've collected it, you can usually see enough, and all you need to see is really the tips of the galea. And, you know, that's, you know, most Andrena.
And most collectors are not, not pulling the tongues. So let's see if we can see enough here. I don't know if that's good enough of a shot. Went down a little bit. Yeah, I think we're in that area where the legs are gonna, I can tilt it more, but it will then hide it even more.
Yeah.
So I'm just gonna say let's not bother. So,
Right.
But Mike, so when you know, a tricky thing is what is pointed?
Yeah,
is it
good
like
point.
Yeah, and what is blunt? So sometimes in some bees a blunt tip can actually be a pointed tip in others because of you know, the relativity of this. So when you're looking at it, when you say pointed, is it like ouch, or is it angular, or is it just in comparison to other Andrena?
You know, LaBerge called it spear-shaped, so that's a little more specific, because, you know, something that's blunt can have its little point, but these taper. These are narrow. They narrow and they taper to a point, and rather than being, and if you look at almost any other Andrena, you know, the galea are more rounded, more broader. They don't taper, and so I mean, it's distinctive, and it's just one of those things that if you can see it, that's it. I mean, that's all you need. But if you can't, then the bluish cast that Sam showed you is helpful, and then the overall smoothness, lack of punctures on the tergites, and these are small. I mean, Cecilia and Polemoniae are small bees, 7 to 8 millimeters, about that. So all those things together, and if you have a flower record, that's suggestive too. So, but what LaBerge said in his revisions of Rhacandrena, Euandrena, and Thysandrena, all three of those, he's basically, the first paragraph starts out with, these are difficult to characterize. So I know we said that last week, but I just want to emphasize it because these are challenging ones. It took me a long time to really learn them.
Yeah, there's several other species to think about when you have a metallic sheen. They tend not to be bluish, more greenish. So nigre and personata all have that little tinge, and so it also gets tricky. Like, am I seeing a metallic sheen or am I not seeing metallic sheen? Is that soap? So yeah, this is, you really do want to look at a lot of specimens, particularly if you can get to a museum, just to get your mind picture around these ideas of distinctly bluish, right? So distinctly bluish could be like you know, as blue as the sky, or it could be like a glimmer of blue, which is more the case here. So even Geranii is not like as blue as an osmia. It's approaching it, but yeah, it's blue for an Andrena. How about that?
Yeah. Okay.
And the one that always gets me is, and Wally LaBerge like to use this a lot, is Andrena barbara. it has a slight purplish sheen, but oh, is it so subtle? And it's
Oh,
not
man.
really, it's a sort of a, yes, it helps confirm, but a lot of times I'm like, do I see it or do I, I keep looking for it. And sometimes I believe I can see it and sometimes not, but it has to be basically a pristine specimen. Anything that dulls it down from poor processing or just age, and it's that sheen appears to be absent.
Yes, comparative. I mean, the more Andrena you see, the better you get. That kind of sounds obvious, but it's a big group. So the rest of the key, we're really dealing with just individual species within the subgenera, with the exception of Thysandrena, because of the fact that they're all so similar. So one of the big breaks between these last couple of couplets is, are the tergites smooth without punctures, or are they sort of punctate? And again, lighting can make the way you see it and the way your lighting strikes the specimen can make a big difference. And sometimes rotating the specimen, moving the light around just to make sure you're really seeing what you think you're seeing. So Euandrena algida and Thysandrena. Oh, Jeff, go ahead.
So I was just going to, and talking about lighting, a lot of times people get these circular LED lights overhead, and they're very good uniform mechanisms. But I find, particularly because of these subtleties like the tessellations or the fine printing that you might find on many parts, looking at the rugose patterns, looking for pits and other kinds of faint features, that taking some of these, I'll call them little LED gooseneck lamps, you might get them at IKEA, which we do, and putting, and the important thing is to diffuse the light. So like the light I have here, I am shining the light through vellum paper, but you can also take like tissue paper, or I use, what's that thing that's in every lab? What is it?
Kim wipes.
Kim wipes. And just rubber band them over the front, so you decrease the glare and you create a more diffuse light. And I know that Jason Gibbs is a huge fan of taking very large fluorescent bulbs in a lamp, which is very clunky, but works for the same reason. So anyway, just thought I'd throw that in because
totally. I mean, the lighting is every bit as important as the quality of your optics in your scope. I mean, bad lighting can make a great scope not work well, and vice versa. So everything Sam said, take to heart, because diffuse light, at least from a lot of these smaller species, things look totally different under that diffuse light than if you had a standardized direct light. So.
Okay.
And this
Sorry,
is
Mike,
no, that's perfect, and that's a perfect place to talk about it, because it makes all the difference in these next two couplets. So Euandrena algida is a northern, at least in the Midwest, it's a northern species. We hardly don't see it unless you get up in the Great Lakes region in north. And I don't know about, probably in the mountains, Sam?
We don't get it in Maryland at all. I guess it's possible in the Appalachians, but it really seems like New England is probably the nearest.
Okay. So that's, I probably should have put that in the key, the geographic component here. So this Euandrena algida is, you're not going to see it unless you're basically up north. But the next choice in the couplet, Thysandrena, the common species is Thysandrena bisolicis, which is, at least in the Midwest, it's fairly common. And it's a willow specialist. So it's geographically, it's not entirely separated from algae, but it's more southern. And again, it's a willow bee. And the two are very similar when you put them side by side, but the facial fovea is different. And lighting can affect how you see that, how you see the fovea as well. So in algae, the fovea don't extend below lower margins of the antennal sockets, but in Thysandrenas, all the Thysandrenas, they do. So it's, again, that's a subtlety, but you know, in practical terms, in most of the regions where you all are working right now, unless you went north, you're not going to find algida, you'll find Thysandrena.
I think algida, again, I don't get to see it very much, but in looking at puzzling over specimens, and every once in a while, it's something will key to it. I'm going like, is it really? I have this impression that it has a fair amount of black in the on the face. Is that correct?
Black? You mean black
Black
hairs?
hairs. Yeah.
Yikes, Sam, I can't remember.
Okay.
You know,
All right, then it's probably not.
we could. Well, no, we should look. I mean, it's a widespread species in the north part of the continent, but there aren't any subspecies, I don't think. But let me make a note of that here. Okay. Okay. The last couplet. Callandrena reappears. So remember, Callandrena we covered weeks ago. Huge subgenus, all specialists on some kind of Asteraceae plant or plants, and they all have these short galea that don't exceed the tips. I mean, short maxillary palps that don't exceed the tips of the galea. The exception in that group is krigiana, which is it doesn't look in terms of mouth parts like a Callandrena. The maxillary palps are longer. I'll just call them normal. They look like a normal length for an Andrena, and it's a krigia specialist. And so that's always a helpful thing, and it's pretty distinctive bee. Very shiny tergites, and the facial fovea are super short and almost you know, kind of triangular in shape, and they just don't extend far at all. And once you see that, I mean, it's very unusual for at least our eastern Andrena to have a species that has such incredibly short facial fovea. So that's pretty significant if you can see that, and usually you can.
So do you find this in very, so I think of krigia as very, so we don't see it. So there might be a couple records, but
Okay.
do you see it in these very dry, the places I associate the plant krigia tends to be dry, very short grassed, can be lawn even kinds of environments, roadsides, things like that, or am I misplacing that? Where do you?
Well, yeah, it's a very natural community type plant. There are at least three species out here, and they're not weedy at all. I mean,
Okay.
unless your road goes through a glade or something. But one of the species is in woodlands, and it's along stream sides, and these are beautiful beautiful flowers, kind of a yellowish, orangish, bloom in the spring. But then we have some real tiny ones, real super tiny. They grow in sand.
Yeah.
And that's probably the ones you're thinking of.
Yeah.
And so it's a diverse genus and diverse habitats, but always what I would call natural, fairly high quality habitats. And so the bee is.
So the bee is restricted too.
Yeah. Yep.
Okay, you're not finding it in lawns.
No.
Okay.
No.
Okay.
Unless you have a really cool lawn,
Right.
But,
well, I mean, some of these areas that I'm thinking of that have it are basically mown but never tended, let's say. So they're just really crappy lawns, but they're on Patuxent, for example, and so it blends pretty easily into these neat plant areas.
Yep. Well,
it's just gravelly.
Yeah, that sounds. That bee might be there, Sam. I don't know. I mean,
I'll have to I'll have to target krigia more than I have. The ones that we have are so small that I just pass over them basically as, some hawkweed kind of thing that I'll just pull a Halictus legatus off of or something.
Right. So,
but it also doesn't show up in bowls in those same areas. That's the thing.
Okay. Yeah, and I have collected
I would expect it.
I've collected them in a lot of bowls. Actually orange, orange bowls, which are similar to the flower, which when you think about it.
Right.
Yeah,
So let's pause for a second and ask, why are you using orange bowls, and do you have to catch other bee species in orange that you don't catch in the other colors?
Well, krigiana was one that I caught. I mean, occasionally showed up in yellow, but I mean, when I first put out the orange bowls, it's like there were like 15 or 20 of them in there, and I thought, so whoa. And now I haven't used orange for a couple of years. I don't know why, but yeah, I don't know. Experiment with colors. If you're, I know it's the standard is white, blue, and yellow, but
okay, yeah,
who knows?
We'll do it. It's a good student project, the Orange Bowl project.
So the other option is on the couplet is Euandrena nigrihirta, which again is a northern species, and the ranges of krigiana and nigrihirta don't overlap, and so I mean that's a geographic characteristic. And nigrihirta is again, if you go in the Upper Great Lakes region, you'll find it, but we certainly don't get it down in the Midwest much. I don't know. It's probably in the mountains, I would imagine.
Yeah, we have it there.
Okay. And then finally, and I don't think this is unique to the Ozarks, but there's still a number of Andrena that are undescribed in our region. And so if you have something that just doesn't key out or doesn't fit you may have something undescribed. And it's just shown up in the last few years. The males are very distinctive. They have a malar spine, which is unique in our
dramatic.
Yeah, dramatically so, right. You can't miss it. And and it's big. It's 10, 11, 12 millimeters. And yet no one has ever seen it until these last few years. I can't explain that. And we even found it in southern Illinois, which is right, Wally LaBerge's backyard in a sense. And the females, which we have figured out, are also distinctive. They have a peaked vertex that's kind of unique. And they both have a pronotal ridge, very distinctive pronotal ridge. So we have records from Georgia, from Indiana, and Illinois, and Missouri. So this is a widespread undescribed species. So, the female you would think is an Andrena sensu strictu. So anyway, keep your eyes open, and not only for this one, but for a lot of others. So just because you can't identify something doesn't mean you can't identify it. It could be that it just hasn't been described. So especially if you're working in unusual habitats or unusual places. And I don't know if Sam, if you've got any in your part of the world that are mysteries.
We've set them aside, but I haven't, I can't describe any. But you mentioned this Cornus one, and I recently was looking at some, and I just was like, this is not keying out to any of the known Cornus specialists. So I'll have to. Those get accumulated as Andrena interesting. And so we meant to go into the Smithsonian and look at the collection, but a lot of that's going to be coming here soon, so we'll punt to that.
Yeah, that's interesting to hear. Okay. Well, that sort of concludes the Eastern Andrena subgenera as best as we could. There's still lots to talk about, and the key needs additional characters, needs to be refined more, but certainly appreciate any feedback or any comments, because that's how it gets better.
That's important. Yep.
Totally. Yep.
So should we do an introduction to the male guide? I don't have a watch on, but I think we have.
Yeah, we
have.
Yeah. Chat. Maybe while Sam opens up this other document, if anybody has any questions, you can just unmute.
They obviously know all the Andrena subgenera. But there's only 500 species in North America.
So with the males, you're gonna with at least with this key, you're gonna notice some similarities to the female key. And right off the bat, the first thing I look at, if I've got a male, and usually it's pretty obvious, the males are the scopa are not there, and et cetera, et cetera. So the males are very easy to separate from the females. First thing I look at is the pronotum, and does it have a ridge or a humeral angle? And unlike some of the females, in most males, if it's present, it's pretty obvious. There are a couple exceptions, but in general, it's a little more boldly displayed in the males, and sometimes dramatically so, than in the females. So that's a good, just like with the females, it's a good way to separate a bunch of groups from other groups if that ridge and or the angle's present. And so if it's present in females, it's going to be present in males. So there's not, there's really no instance of of a male that's got a really dramatic dorsiventral ridge on the pronotum, and the females are all simple. So there's a lot of similarities there.
Mike,
Yeah.
Do you think that this pronotal ridge, and which is often just notched out, has something to do with pulling the legs up tight against the body, as you might see in the pleura and some of the other species groups?
I don't know.
Yeah.
You mean when they're mating? Or what?
No, well, not necessarily maybe mating, but just as like to in the nest or hunkering down. It seems like that's an area that allows the body to be more compressed if you can move your legs in and closer to your body.
Yeah.
But
yeah.
Again, maybe not.
Yeah,
I'm just trying to figure out why.
Yeah.
All right, something to think about.
Yeah. So that we're gonna we're gonna cover, basically it's the same groups in the females that we covered, but the males are they're different enough so that in most cases the characteristics that you use to identify males are quite different from those that you use to identify females at the species or subgenus level.
So Claire wants to say something.
Go ahead.
Eric asks, do males emerge first during their time periods of being seen throughout the year?
Did you get that, Mike?
Yeah. In general, the males are what we call protandrous. In Andrena, the males come out first, and hope to find a freshly emerged female. And I'm trying to think of exceptions, but in Andrena, but I can't think of any. But it's not dramatic. I mean, they don't emerge like weeks ahead of time. It's usually a day. You see the males first, and then the next day or two you see the females. At least that's been my experience.
An odd thing I see, Mike, and I don't know if you see this too, is that sometimes they group or maybe even lek. I'm not sure, but they'll be just on vegetation, and there'll be a whole bunch of them, in sometimes multiple species in one area, and I can't figure out why.
Yeah, I've definitely seen the groups of males, but I never, never more than one species. But you often see them, they're going to be where they're going to look for virgin females is at the nest site. And so a lot of times you'll see them cruising over the ground, and also a lot of nomada mixed in. But that's where the nest, that's where they're going to be there's a nest aggregation or nest sites. And the males know it, parasites know it, and that's a good way to find nests, actually, is to follow those, both the males and the nomada. So if you've got a male and it's got a dorsiventral ridge or a humeral angle, the next thing.
One more question. Do Andrena overwinter as adults or larvae?
Post-feeding in the Midwest, what we call post-feeding larvae, what used to be called pre-pupa. So they've eaten everything, and the next step is to pupate, and I'm trying to think exactly. There might be some that overwinter as adults, but I'm not in the Midwest, but I'm not aware of it. It's, as far as I know, it's always prepupa. And then there's some, some trigger, and it's always been a mystery to me. A bee that's underground, a foot, maybe 2 feet underground at the most. How do they, what are the signals they get from the environment that it's time to get on with it? It's kind of pretty amazing.
Even the early spring bees don't transition to adults in the fall.
I should know that off the top of my head. Now you've made me wonder. We tell you what, I'll double check the references on that to see if there are some exceptions, and we'll let people know. I've dug up some erythrogaster and perplexa in the fall, and they were prepupa. So
okay.
But I mean, with a group that big, with hundreds of species, I wouldn't be surprised if there's some variation in there, geographically as well as just genetically. That was a very helpful answer, but we'll find out more.
I think it's very helpful.
Okay. So the next thing, and this is really a conspicuous thing with a lot of male Andrena, the clypeus, or the face in general. It can have yellow or ivory macula. And this is really important because it helps separate a lot of species from other species. And it's obvious, very distinctive. So, and the patterns are different in different species. So that's a biggie. If you see yellow or ivory macula on the face, and sometimes they can be very big, very conspicuous, cover half the face practically. And in other cases, they might just be a few dots and everything in between. So it's an important character, but it really varies a lot. And sometimes if you're not looking carefully, it's easy to miss. We'll talk about Andrena bioli later, but that's one where sometimes the clypeus is yellows, or got a few dots in the paraocular areas, and other individuals it's completely dark. So there occasionally is variation. And so some of these things are going to come out twice in the key because of that. But this is a good one, very conspicuous. If you've got a bee with the pronotal ridge and the clypeus or the face has some yellow, boom. There's only a relatively small number that fit that, and several of them are rare, and a couple of them are super common. So the first one, and this is one with we've had some interesting developments the past year or so. When you look at the side of the cheek, the gena laterally, there are two species that it comes to. It's like a big angle sticking out, and one is Banksii, which it's not dramatic, but it's clearly there, and the other is Cerebrata, which is like it's just weird. I don't know if you have any of those, Sam, but pretty cool.
Not the male.
Okay, but it's dramatic and you can't miss it. And the Cerebrata, it's starting to look pretty conclusively like it's a specialist on mock orange Philadelphus. We've found recently, people in Tennessee have found females collecting pollen from the plants, Philadelphus, and males sleeping in the flowers. So, if you've got any Philadelphus, even if it's in a landscape plant, it doesn't have to be out in nature somewhere. Look when it's flowering, look at it carefully, and you might see these bees because they, heretofore they've been considered quite rare, but maybe they're not as rare as we thought.
And they're small.
Yes, they're small.
I'm sorry.
Face.
What did you say, Claire?
Click to see the species page of the Cerebrata because it has the thing on the face and it's a diagram.
Did you say Banksy was a in that group?
Yeah, it is.
Or, okay, I'll have to go back.
Wait a minute. I may be mixing. I'm sorry. I get those.
There's a Mackie here.
Yeah. which I'd never seen.
I mean,
It's
but
Western.
Banksy does have the male yellow clippius and big
Right. Big
hook and
pink.
knob on it.
Yeah, yeah. And that, I don't know if you got a picture of that one.
Yes. Let's see. It's the easiest way to go there. So yeah, there's the male with the yellow. Actually, I can't see. I don't think you can see the knob very well in this picture. Oh, you can. There it is, but not extremely.
Yeah,
But it's a big projection.
oh, there you go. Yeah, that's good.
Yeah.
Yeah.
So, but the interesting thing is a lot of times you'll look at this bee face on, and it's like, oh, what kind of yellow-faced bee is that? And you actually initially miss the fact that it's got that big cheek. See, that looks like, oh, that must be Miserabilis or something.
Right.
And then if you don't flip it on its side, that knob isn't necessarily the first thing you see. So many things. But I don't have a Cerebrata picture.
Yeah, that was a bee I never saw until the last few years, and I've since seen bunches of them. So I think it's a case of one of those things. Once you know where to look,
we see it regularly, and often associated with willow, but I don't know that I would call it a willow specialist. But a lot of times we'll pull in specimens from from collecting on willow, but they also show up in other places. So I hesitate to call it any kind of willow specialist.
Well, it may not be exclusively a mock orange specialist either then. So
No, I'm talking about Banksy.
Oh, Banksy. Okay, yeah. Well,
Yeah,
no,
Banksy.
Sam, that's the only place I've collected it is from willow. So
okay.
I mean, so in the Midwest, so maybe it is. I'm sorry. Yeah.
Yeah, okay.
Okay.
All right.
Yeah. Okay. Yeah. Thanks for the reminder. Yeah, Banksy is in. Well,
We can figure it out.
Yeah, we will. So, Cerebrata, very distinctive, and you're not going to see anything quite like that. So, another very important characteristic in this group is the base of the mandible, ventrally, what's called ventral basally, has a pronounced tooth or an angle, and sometimes it's quite dramatic. And other times it's a little more subtle, but it's again a very helpful characteristic, and not just in this group, but for others that we're going to see later, that can be pretty helpful and usually easy to see. Even the mandibles don't have to be open. You can see it no matter what shape the mandibles are in. But in those cases where it's a little subtle, you can sometimes miss it.
Yeah, I usually find that it's not subtle, but the females, which Wally LaBerge in his keys will often say have a tooth, and it's like, come on, really?
Yeah,
it's so subtle.
I agree completely.
Yeah. All right. Good. I'm glad someone else
Yeah,
does.
yeah. That's why I just don't see it, honestly.
Yeah.
And so anyway, so if you've got this ventral basal tooth angle on the mandible, next you go to couplet 5, and there are only 2 groups in this section of the key that have that, and they're both, pretty rare. And one of those we already talked is Banksy, and no, it's not. I'm sorry, it's Dimorpha.
Yep.
Is one. Yes. Okay. Names get confusing. Rhacandrena, and I've only collected a handful of these over the years, but the males have a very distinctive, smooth, shiny pygidial plate, which most males do not in Andrena. But these are conspicuous, and it doesn't look exactly like the female pygidial plate, but close. And again, there's no hair on it. It's very shiny, triangular. And not, you know, sometimes the abdomen is contracted and you might not be able to see it, but boy, if you can, I mean, you immediately know what group you're dealing with. And I'm trying to think of some of the other characters. That's the most outstanding one for the Archaeandrena. And then Derandrena, these are small. The Archaeandrenas are pretty big bees, 10, 11, 12 millimeters. But Derandrenas are small, and they're all specialists on something, and often considered, I mean, I think of them as rare. But Uvularii is one, and I know we talked about whether it really belongs in that subgenus or not, but for the present, we're just going to say it does to make things simple. So Uvularii is on the genus Uvularia, and then Zizioformis, Andrena zizioformis, the males look very, very much like Uvularia, is on Potentilla. And these have the big, long mandibles with this strong tooth on the ventral basal margin, very distinctive bees. And bees that most people probably never see. And in fact, the male is still of Uvularia is still undescribed, technically.
Didn't Milan describe it, or am I mistaken in that?
Maybe I
think
that.
I think they collected it, and you know, pulled genitalia and all kinds of stuff, but maybe they maybe they didn't get published yet. Oh, yeah. I wonder why it's not in like the Bees of Massachusetts. Okay. Yeah.
Anyway, these are specialists. You got to find their plants to find them. Have caught them in yellow bowls a lot.
Yeah.
So
they're right down there at the same level as the Potentilla. The Potentilla, I think of them as just the common
yes.
Potentilla.
Yeah.
And it can be in a lawn situation, but a lot of times it's in a poor soil, and they're just this big mat of them. And if you spend some time sitting parked there, you can see them, you know, flying around.
Yeah.
And then get the Panurgus off too.
Okay. So those are good ones to look for. So if you got a male with the pronotal ridge, yellow clypeus, or some yellow on the face somewhere, and without a ventral basal tooth or angle on the mandible, you know, we go to couplet 6, and here again it's another pretty much straightforward case. Either the forewing has 2 submarginal cells or it has 3. And like Sam has said before, look at both wings, and sometimes strange things can happen. But in general, this is a pretty much take it to the bank situation. So this is the subgenus Parandrena, and again, these are willow specialists. There are only two in the east, and they're in the Midwest. If you find willows, well, there's actually, I think there's a third too. Is it there? Yeah, there is. There are 3, and all of them are willow specialists. They all have just the 2 submarginal cells, and I mean, that pretty much nails it right there. I don't know how common they are in the east, but in the Midwest, often in sandy areas, but I don't know that they're entirely sand obligates. Wellesleyana probably is. That's the only place I've ever found that species is in sands.
Right, gravel pits.
Okay.
Our sand mines up north.
Yep.
Yeah. So we don't get that one. And the other 2 are super rare.
Wow. Okay.
You know, and I'm, you know, wherever, if there's a willow blooming, I'm going to be collecting off it, but they're not really a major part of the willow fauna.
Okay. Yeah, and in the Midwest they are. It's pretty much, you find willow, you're going to find Andrenoides, probably, or Nida. So that's it's a pretty straightforward group, the two submarginal cells and the yellow calycus and the pronotal ridge. That's it. So, and I didn't mention this, but the sixth sternum, and this is going to reappear in other groups, but the sixth sternum is reflexed. And what that means is if you don't look at the underside of the bee or look at in lateral view at the end of the abdomen, you can miss it. But it's an important feature in some various groups of male andrenas, and we'll encounter it again. And so what we mean by reflexed is it's sort of, instead of being straight, the tip of it is bent. Often has teeth or angles on the lateral margins. Sometimes it's real obvious, but sometimes it can be gunked up, and then it can be difficult sometimes to recognize. But it's a very important feature, and that's one of those things. When I'm looking at male Andrena and nothing jumps out at me, one of the things I always look at right away is that, well, what's that sixth sternum look like? Because that that helps, very helpful. So
yeah, and that term is always tripped me up because there was no one there to ask. So reflexed, what does that mean? And so it's just not a commonly used term anymore. And then also, there's an orientation. You're not usually looking upside down at the bee. You've turned the bee over. So it's really, in some ways, in the view that you're seeing, it's like the whole end is just bent up, usually at a right angle across the margin of the very end of the rim of S6. And yeah, it is. I use it all the time now, but initially when I was doing that, and a lot of times this is not the most common character, but in some groups it's all over the place. But if the average Andrena doesn't have this, nor does any of the other groups. So that's why it makes it special. So if you're if you're looking for it in a place that it doesn't occur, it's really frustrating. Is that reflexed or not? And then you run across a couple to view.
Yeah, and there are a couple super common ones that have it that we'll get to eventually. So yeah, the next choice, couplet 7, is one, at least it's one of the most common Andrenas in the spring in every place I've ever been, and it's the subgenus Euandrena, which now the name's been changed, but I'm not going to get into that right now. The species is Cresnii, and there's only one. That's Euandrena cresnii. And there are a couple subspecies, but the one in the east is Cresnii. And usually the males have an incredible, half the face is yellow or ivory. I mean, you just can't miss it, even with the naked eye. But it does vary, and I've seen a few individuals that have no yellow on the face at all, which drove me crazy until I looked at the genitalia, which are very distinctive. So there's some variation in that bee, and but it's super common. And if you're going to be collecting Andrena in the spring, you're going to find this bee, and it visits a lot of different plants, and is in all kinds of habitats. It's my experience. It's one of the most common Andrena in the eastern part of the country. I don't know if that's true where you are, Sam, but
completely. And in lawns and
okay,
and disturbed areas.
Yep.
And and every once in a while you can get a male that has almost, you may have mentioned this while I was futzing around here, you can get a male with almost none of this yellow.
Yes.
Like it's all eroded away.
Yep.
And they're like, what am I looking at? This must be new, but it happens, one out of, maybe a couple hundred, maybe more than that, maybe 500 specimens. And a lot of this will be gone.
Yeah.
And, but usually there's shadows of it, and it patches up. I don't know why.
Yeah. And because of that, this species comes out in a part of the key later on, where we're talking about males that are altered, just because of that one in 100 chance. But it happens, so
yeah.
Why, I don't know, but
and
so
really I always note this really wide head.
Very much. And yeah. Yep.
Oh, so Claire.
Cool. Oh, time.
Claire's giving us a timeout.
Okay,
Yeah, we just got a memo that you need to be on at 2 o'clock, like on the dot on time.
Me?
Yeah,
okay,
we got passed a note.
Okay.
okay.
I'm gonna wrap us up. So any parting words on that whole Andrena? I'm sorry if I interrupted the Crestodonii at the end of that. Was there anything else you wanted to make sure was finished being said?
Not me.
Yeah, distinct V. You can just look at the face.
Yeah, in the field, you don't even need a microscope. Okay,
All right. Apparently I'm
Yes,
on
you're-
call, so
okay,
we have to leave.
okay. Thank you, Claire.
Okay.
Thank you.
Thank you, guys.
Okay, all right.